Hello everyone, we're just going to wait a few moments to let some people enter into the webinar and then we will start. So I will begin. So hello and welcome to the Institute for Molecular Science and Engineering webinar, Nanoparticles for Treatment of Tuberculosis Meningitis. This is going to be presented by Professor Alexandra Porter and Professor Alexandra Porter and Dr. Alexandra Pina from the Department of Materials at Imperial College London. This webinar is part of the Molecular Level Understanding is Crucial for Targeted Drug Delivery in the Brain series. I'm Leah Adamson and I'm the Events Officer for IMSE. I'd firstly like to apologise that you cannot see me. Unfortunately, my camera is not working. So we're going to be putting some helpful links into the chat function. This will include links to the IMSE Eventbrite page where you can find the registration for all of our upcoming webinars, including all of the webinars for this series. The IMSE YouTube channel where you can see all of our previous webinars uploaded, our IMSE blog, the Never Lick the Spoon podcast, all of our previous briefing papers and the registration to our newsletter, which you can keep up to date with all of our news and events. So just a few top tips to make sure that we can have a really good webinar session. Please make sure that your microphone and video are switched off. If you wish to ask a question, please put this into the Q&A function at any time during the webinar. Your questions will be answered at the end, but please do not put your questions pertaining to the presentation into the chat function. If you have any technical difficulties, then please message IMSE Imperial privately in the chat function and I will try and help you where possible. So I'm going to now introduce our presenters for today. So Professor Porter's group uses correlative electron microscopy techniques to characterize interfaces between biomaterials and cells. Her great interest is in developing a mechanistic understanding of how the chemistry of these materials controls their degradation behaviour and ultimate bioactivity to improve their performance and safety. She works on a number of systems, including the environmental fate and toxicology of nanomaterials, plastics, and how bones' qualities and toughness become altered in pathology and pathological issues. She also designs new nanomaterials to cross the blood-brain barrier and to treat Parkinson's disease for the treatment of tuberculosis and breast cancer. And Dr. Pina is an Imperial College Research Fellow for the Department of Materials. Her research interests focus on design, synthesis, and characterization of inorganic nanoparticles and nanocomposites for biomedical applications, in particular the interest in drug delivery and antioxidant systems. The nanocomposite particles are used for the treatment of neurodegenerative diseases, osteoporosis, cancer, and tuberculosis meningitis. So I'd like to say thank you to our speakers, and I will hand over to them now. Thank you very much.
Okay, so thank you very much for inviting us to give this talk. Our talk today is on inorganic nanoparticles for treatment of tuberculosis meningitis. I'm Alexandra Porter, and this is Dr. Alexandra Pina, who's an Imperial College Research Fellow who works between Imperial Materials Department and the Crick Institute. That's the next slide. Okay, so why are we interested in developing new nanoparticles for delivery inside the brain? So when I started this work, we were interested in neurodegenerative diseases, and the options for treatment of these diseases are very limited. And one of the major issues here is that there is a lack of neuroprotectants available. And if we look at the structure of the brain on the right over here, it is has this network of brain endothelial cells around the brain. And these brain endothelial cells, so you have the blood and the brain, and they form, there's a barrier that's formed between these cells and the brain that's called the blood-brain barrier. It's a highly protective barrier as it needs to be. But this creates a major challenge for drug delivery and makes it very difficult to deliver drugs across this barrier from the blood into the brain. As an aside issue, which is not relevant to this talk, there's also a lack of neuroprotectants that can protect neurons with minimal side effects. So there's two major issues here. And in fact, as was highlighted by Daniela Dini in his talk, only 5% of drugs are able to cross this blood-brain barrier. So there's an urgent need for new drug delivery tools, mechanisms to deliver these drugs inside the brain. And in the case of Parkinson's disease, target these damaged neurons and treat them. Next slide. Okay. So the approach that we were looking at was whether or not you could use nanomedicines, so nanoparticles, to attach the drug onto these particles and use them to deliver the drug from the bloodstream across these brain endothelial cells of the blood-brain barrier and into the brain. So what's the advantage of using nanomedicines? So it's threefold. So first of all, because of their small size, they have a very large surface area to deliver significant cargoes of drugs across these brain endothelial cells into the brain. Several of the drugs are quite insoluble, and by reformulating, encapsulating these drugs inside the particles, you can overcome solubility issues. And also they're very small, so they're small enough to circulate systemically in the bloodstream. So here you have an example of the different types of nanoparticles. They're about 100 nanometers or less in diameter, so they're small enough to be internalized by cells, in this case the brain endothelial cells, and possibly translocate across these cells into the brain. And there is evidence from in vivo animal studies that they can be used successfully. So here's an example of a nanoparticle that was conjugated with a large amino acid transporter targeting group, and they showed when they injected the nanoparticles bearing this group into the circulation that they were able to accumulate inside the brain, compared so more so than the control particles. Next slide. So because of this potential for using the particles, we focused then on whether or not we could use this as a strategy for delivering therapeutics across the blood-brain barrier. So here we see an example now of the blood in the middle, and you can see the brain side. And then the question was, can you IV inject these particles into the bloodstream and use them to deliver these drugs across these brain endothelial cells, that's these barrier cells, into the brain, where they would interact with different cells on the brain side, and that's what Alexandra will talk about. So here was what we were hoping that the particles would cross. So when we started this project quite a few years ago, we started with what might be quite a naive approach, but I was working more on the toxicology of nanomaterials, and there was a big question around whether, next slide, whether carbon nanotubes that were being developed for different applications, but also for applications in medicine, could cross the blood-brain barrier, causing adverse effects. And so I had evidence from one of the first studies that I worked on when I came to Imperial. So what we did was to expose epithelial cells, in this case, to carbon nanotubes, short carbon nanotubes. And we found that they translocated, you can see in this tomogram here, dark field tomogram, translocated across the plasma membrane into the cytoplasm of the cell. So we set up the hypothesis that if they can translocate across this plasma membrane, could they then diffuse into the cytoplasm of these cells and across the blood-brain barrier? So I worked together with Professor Milo Shafra in the Department of Chemistry and Materials, and what he did was to use these carbon nanotubes as a starting material, and he shortened them, so they were around 700 nanometers in length, 30 nanometers in diameter. And then what we did was to graft them with different functional groups so that they had, so P4BP, so positive charge with the PEGMA, neutral charge, and PMMA with a negative charge. So we wanted to understand what the effective charge was on crossing. So next slide. Okay, so here's what we did. We used an in vitro transwell assay, and we applied the multimole nanotubes to the apical side, that's the blood side. We cultured these endothelial cells on a transwell, on an insert, and then we measured, as a function of time, accumulation of these multimole nanotubes in the basal side of this transwell. And this is the method that we've used throughout the work that we're presenting today. And what we found, if you just focus over here, I won't go through all of the data, basal, C, and T mass, we found that the negatively charged carbon nanotubes cross more significantly than the positively charged particles. So this is this SP-MAC, they cross more significantly than the positively charged and neutral charged particles. And we measured that using the UV visible spectroscopy, and we also quantified this using electron microscopy. Why? So we didn't get fully to the bottom of it, but one of the reasons that we found was that the positively charged particles get trapped within the cells, so they're not able to cross significantly the blood-brain barrier. So next slide. And then we confirmed this using transmission electron microscopy, where we took images of the different particles, negatively charged, positively charged, neutral charged, crossing the blood-brain barrier. And for example, in Q on the bottom right, you can see here at the basal side, a nanotube which has crossed to the basal side across these cells. And again, we were seeing at the apical side evidence of this transcytosis mechanism, so the insertion process where the nanotubes were able to insert into the membrane of these cells. So next slide. Okay, so because we were seeing quite small amounts, we needed to find a better strategy to deliver these particles and eventually drugs across the blood-brain barrier. So what we did was to look at the different mechanisms by which molecules can cross the blood-brain barrier. So here's our blood-brain barrier, a cartoon of it, and there's a number of different pathways by which different molecules can cross the blood-brain barrier. So I've listed them out here. You've got passive diffusion of lipid-soluble non-polar substances, carrier-mediated influx of polar substances, small peptides, but the interesting one is this receptor-mediated transcytosis of certain proteins, for example, insulin transferrin. And there's been quite a bit of work showing that if you take a particle and you functionalize it with these groups, you can increase the amount of crossing of these particles across these brain endothelial cells by piggybacking these mechanisms. Next slide. Okay, so here's what we did. So this was working with Professor David Dexter, who's now head of research for Parkinson's Research UK. So we tried to get these particles across the blood-brain barrier with limited success. But what we trialed was this. So our idea was that if you conjugate L-Dopa onto the surface of the nanostars, maybe this could promote crossing. So why did we select L-Dopa? It's an FDA-approved drug, and it's used to treat symptoms of Parkinson's disease, and it is known to cross the blood-brain barrier. How does it cross? Well, it's thought that it is transported by this large amino acid, LAT1 transporter, and that this is highly expressed at the blood-brain barrier compared to non-peripheral barrier tissue. So this was our hypothesis, that if we coat the particles, in this case we were using gold with the L-Dopa, we were able to improve crossing. So what we did was to use gold nanoparticle seeds. We mixed them together with gold salt and the L-Dopa that you can see here, and then what we found was that instead of making particles, we made these flower-shaped materials that you see over on the right. We call them nanoflowers. They could be called nano-urchins, nanostars. And so this is what we made. And then next slide. Then what we did was to use our Transwell assay again, and we placed nanostars, L-Dopa nanostars, in the apical side, our blood side. And then we measured the amount of crossing into the basal or the brain side of these cells, of the Transwell. And we were using two different cell types. We were using primary brain endothelial cells from a murine model, and also a human-derived cell line. And this is what we found. With our TEM, so with our transmission electron microscopy, when we took sections through the brain endothelial cells, we found them at the apical side and some evidence of them at the basolateral side. We also looked inside the cells, and we found, this is the red here, evidence of these particles within these cells. But on the bottom right, this is what was interesting. So this are the two cell types, these human-derived cell line and our primary cells. And what we found when we compared our L-Dopa Nanostars to, this is just a catechol for EC, that's our control, we found significantly more crossing of our L-Dopa Nanostars across both cell types, but significantly for the primary endothelial cells. And you can see that the amounts we were getting, 60% or so, was much higher than for the carbon nanotubes. So this looked quite promising. Next slide. The question is, why is this an effect of the L-Dopa, or is this an effect of geometry? And Alexandra will talk more about this, but we have shown using electron microscopy that the stars again are able, it seems to penetrate into the plasma membrane of these cells, and we have quantified and shown that the length of these spikes is proportional so as you increase the length, you increase the amount of crossing. So there does seem to be a geometry effect as well. Next slide. So now I hand over to Alexandra. Thank you.
Yeah, so thank you so much, Alex, for the introduction and the different application of the nanomaterials to delivery drugs across the blood-brain barrier. So the second part of the talk will focus on the use of inorganic nanoparticles for the treatment of tuberculosis meningitis. So we know that the tuberculosis is known since then 1882, when Robert Koch announced the discovery of Mycobacterium tuberculosis. And despite being considered an old disease, tuberculosis is a leading killer of people, especially people with HIV, and it's also a major cause of death related to the antimicrobial resistance. So in 2018, if you can see from this picture on the left, the World Health Organization reported that 1.5 million of people died from the TB. So the transmission of Mycobacterium tuberculosis is an airborne transmission. Consequently, the first focus of infection is the lung. But a small number of tuberculous bacilli can enter the bloodstream and spread throughout the body in different organs. So I'm going to concentrate today some results that are related to the treatment of tuberculosis meningitis. So I'm going to concentrate on the central nervous system TB. What happened in the tuberculosis meningitis is that we have like to cure two things. First of all, we need to have like an antibiotics to treat and to kill the bacteria inside the brain. And the second major problem is related to the infection, inflammation, especially the inflammation that we have in the meninges. The meninges, as you can see here, are like a part of the brain that can protect the brain. And in one layer of the meninges, we have, as you can see here, the blood vessel. So what happens is that the bacilli, the tuberculous bacilli, can enter the bloodstream and can spread to the brain. The mechanism through which the bacilli can reach the brain is not fully understood, but Wilkinson et al. Try to explain this mechanism. So as you can see here, so what happened is that we have bacilli in like free form in the blood, and then these bacteria, these bacilli, can be uptaken inside neutrophile, monocyte, or lymphocyte, and they can go and pass and cross the endothelial cells in the blood-brain barrier and reach the brain side. So in the brain side, what happened is that it's gonna, all of these cells together with the bacilli, they are gonna form a rich focus that can be like wrapped and deliver the bacteria inside the CSF and the brain. Here in this video is a real video. You can see the exudate coming from the inflammation that we have in the blood-brain barrier. And this video is a courtesy of the Professor Figagi from the Division of Neurosurgery in the University of Cape Town. So what's the problem with the treatment of tuberculous meningitis? First of all, as we said before, the tuberculous meningitis is a major killer in children and HIV co-infected patients. Then we have to deal with the brain inflammation. And it's really difficult, the delivery of the drugs, including antibiotics and anti-inflammatory drugs inside the brain. And really important is that we are having a lot of antimicrobial resistance. So it's time for action. So how we can act, how we can try to deliver antibiotics and anti-inflammatory drug in the brain. Here we can see a library of nanoparticles that we have. So in this picture, we can see a silica nanostar, which play a role of a carrier of the drug. And inside these mesoporous nanostars, we encapsulated the antibiotics. So we started with the rifampicin, which is an antibiotic that has different problem to reach the brain. And inside the silica carrier, we put also sodium oxide nanoparticles due to the anti-inflammatory and antioxidant properties. So how we fabricate the silica nanostar? So we use a sol-gel method, and by changing different parameters in the synthesis, such as the co-solvent, the surfactant concentration, aging time, stirring rating, temperature, and so on, we are able to synthesize different type of silica nanostars with different sites and different spike length. So using this synthesis, we can control not only the size of the particles, but also the length of the spikes. So here I reported two types of nanoparticles. On the top part picture, you can see a nanoparticles with around 85 nanometers in diameter and with spike length about 9 nanometers. On the bottom pictures, we have the same particles that are around 85 nanometers, but they have longer spikes, which are around 18 nanometers. We also perform some electrotomography measurement, and we can see here in these tomography images the spike of the particles and how this particle also looks like mesoporous. After this step, we encapsulate the two drugs. So the first drug is represented by sodium nitrate, and the second drug is represented by the rifampicin. So we used a diffusion method for both drugs encapsulations. We did like separately and together the encapsulation of these drugs. So we encapsulate silica alone in the silica sodium in the silica nanostar and rifampicin in the silica nanostar, and then we try to encapsulate both in the silica nanostars. So the percentage after encapsulation, we measure the composition of these particles, which is around. We have 68% of Si and 32% of cilium. And also, if we look at the surface area measurement, we can see that we have an increase of surface area when we have the sodium oxide. This is a a bit different respect to other type of drug encapsulation that I have done in the past, because when you encapsulate some drug inside the pores, you have a reduction of surface area. In this case, we have an increase. So it means that serum oxide is most on the surface of the spike. And regarding the rifampicin, as you can see in this table, we try different weight ratio between the silica nanostar carrier and the rifampicin, and we calculate the loading capacity and encapsulation efficiency. So the best results here is with a ratio, 1-to-1 ratio, because we have like 60% of loading capacity and we have an encapsulation efficiency around 55. So when we increase the amount of rifampicin, the encapsulation efficiency is going like to drop. When we encapsulate both cerium and rifampicin, we still have around 68, more close to 70% of loading capacity, but we have encapsulation efficiency. But I didn't report the number, but I can say that in terms of milligram, when we use cerium and rifampicin together, we encapsulate around 130 milligram of rifampicin. On the other hand, when we have only rifampicin, in 1-to-1 ratio, we encapsulate 16 milligram. So the next step was like try to understand if these nanoparticles can have any interaction with the immune system. And what we have done is to see the polymorphonucleated cells that they come from the blood of different patients to see the toxicity of these nanoparticles. And we have also detected some cytokine that I'm not reporting the results today. So what we have done was like take a blood from the patient, and the next results, we show results that are the mean of 3 patients. We centrifuge the blood and we extract the polymorphonucleate cells from this. After that, we incubate it, we plate our cells in the 96 plate and we incubate with the nanoparticles, and we incubate in like at 37 degrees for 24, 48, and 72 hours. So after that, we check using the MTS assay, the cells viability. So what you can see from these graphs here is that going from the lowest nanoparticles concentration to the highest nanoparticles concentration, the nanoparticles are safe, both the one without nanoseria, they are safe until 48 hours. Because after 48 hours, and we can see here in 72 hours, the particle, they start to be toxic, especially at the concentration of 1000 microgram per ml. So after this, we want to see the uptaken of these particles. So if the particles are uptaken or not in the PBMC cells. And in order to do this using a confocal microscopy, we need to functionalize the particles with a dye, which is the FITS. So this is like the chemistry that we used, is like a click chemistry. And on the bottom, we can see the FTIR that report the successful functionalization. So in these images, the confocal images, in green, we can see our particles, in blue, the nucleus of the PBMC cells, and in red, the cytoplasm. And in this video, we can see that the particles are uptaken inside the cells. But in order to be sure, we perform some orthogonal view of these particles, and we can see that we have in all the plane the presence of the particles. So at this point, which is like a next work to do, it will be like the quantification of these nanoparticles. But then we move, since we want to see if these nanoparticles can cross the blood-brain barrier and compare this star shape to the spherical shape, before to build up a 3D model of BBB, we test the viability and the toxicity of the nanoparticles in all type of cells that we use to build up a 3D model of BBB. So we use endothelial cells, astrocyte, pericyte, and microglia. And we incubated also in this time all of these type of cells with different nanoparticle concentration, and we perform TSSA after 24, 48, and 72 hours. So the first results that I'm showing here are regarding the viability cells, especially in astrocyte. So in astrocyte, we can see that all the concentration of these nanoparticles in all the time point are safe, and we can see that they are like promoting, they are increasing the cell's viability more than 100%. This is need to be like studies, why, what's happening in these cells, why is this, why the nanoparticles are giving this benefit to the cells. In the pericyte cells, we can see that both nanoparticles are safe at all concentration, but at the highest concentration, 1000 microgram per ml, and after 72 hours of incubation, they start to be toxic, especially the one containing cerium oxide. In this slide, we can see same experiment, but in the endothelial cells. And also we have more or less the same situation in the endothelial cells of the pericyte. We are safe until 48 hours. And after 72 hours at the highest concentration, the particles, especially the one with cerium, they start to be toxic. Last type of cells are the microglia cells, where the mycobacteria went inside the brain. And also here we can see that these particles are safe, but at 72 hours we have the opposite situation. So the one with cerium oxide, they are safe at the highest concentration, and the empty, so the carrier, the silica one, is start to be toxic. So after that, and after we know which concentration we can take in order to incubate all the cells together in a 3D BBB model, so we choose the 1 microgram per ml concentration, which was like safe for all type of cells, and we start to build the blood-brain barrier. So the blood-brain barrier is represented by this insert. In this insert, on the top part, we culture endothelial cells. Then we have a membrane. And on the bottom part of the membrane, we have a mixture of astrocyte and pericyte. And on the bottom part of the plate and the well, we have a microglia cells. So what we did was like to incubate this model, BBB model, with 1 microgram per ml of nanoparticles, and then try to see what happens after 48 and 72 hours of incubation. And we fix and stain the cells, I mean the whole model. And we took some pictures and we did some analysis at the confocal. So in this confocal video, you can see that our particles are in green because are again functionalized with fits. And we do like the zeta stack. So we acquired different images at different zeta going from the top part to the bottom part of the membrane. So and what we can see here is, here we see these green dots, then here we can see the endothelial cells, the membrane, and then the astrocyte and pericyte. So the first problem that we encountered here was that when we took images of the control, we can see some fluorescence, green fluorescence coming from that samples. So then what we have done so was, before to quantify the particles, was like to exclude all the pixel coming from the control. And then we were able to compare the results between the star-shaped nanoparticles and the spherical one. So in this graph, this representation, so we are like reading the BBB in the opposite direction that we saw before, because the confocal is reading from the bottom to the top. And we can see that the star-shaped nanoparticles can cross better the blood-brain barrier respect to the spherical one. So this line here in red represents the membrane. And by quantifying the percentage of the green dots coming from the particles, we can assess and we can say that the nanostars are better uptaken. They better cross the blood-brain barrier. And also, it seems like that the spherical one, they have some like forward and backward in the blood-brain barrier. So the conclusions is that we reproduce a synthesis to synthesize the nanostar. And we are able to tune both sides and spike length. We encapsulate rifampicin as an antibiotic and serum oxide as an anti-inflammatory drug in the nanostars. Then we show that the nanostar are safe in both blood cells and in both human neuronal cells within the 48-hour and the highest concentration. And so for the first time, we show that in the 3D model, the crossing of the stars, the crossing of the stars nanoparticles is better respect to the spherical one. So there is like the possibility to use this silica nanostar as a carrier to deliver not only antibiotic and anti-inflammatory across the blood-brain barrier, but any drug that has activity inside the brain. So we would like, me and Alex, to thank a lot of people that collaborate to this work. So Alex want to thank Professor Milo Scheffer, Mary Ryan, David Baxter, and Daniel Gonzalez, Fang She, Rosalia Lopez, Angela Good, and Ong Zhan. And I really want to thank all the Imperial crew that helped me to do this experiment for the tuberculosis meningitis treatment, and all the crew that helped me on the bio application of these nanoparticles. And thank you all for your kind attention. So we are happy to take any question.
So I can see there's one question in the chat. Why do the negatively charged nanotubes cross, whereas positively charged particles internalize in cells via endocytosis? We think it's because on the surface of the brain endothelial cells, you've got a glycocalyx layer, which is negatively charged. So that we thought that the positively charged particles are getting trapped within this glycocalyx layer. So when we measured cellular association, there was much more cell association for the positively charged particles, much more than with the negatively charged particles. So the data suggests that the negatively charged particles aren't getting trapped, are not accumulating inside the cells or this glycocalyx, possibly this glycocalyx layer, and so are able to cross.
Yeah, I think that the next question is for me.
Yeah.
So the next question is, can you please explain the role of spike structure? If the nanoparticle shape, Syria-caped mesoporous silica nanoparticles change, but the composition remain the same, will it affect the therapeutic efficacy? So I'm going to reply this. The spike, so the role of the spike structure is like they have a different interaction in crossing the blood-brain barrier respect to the spherical one, because the spiky have less contact with the cells, the endothelial cells in the barrier, and they can like penetrate better respect to the spherical one, that they have like a higher surface contact, and they are like retained more in the blood, and they have like less possibility to cross the blood-brain barrier. So regarding the composition, I think that the composition is the same of the when we have serum, and I don't think it's gonna affect the therapeutic efficacy. So when we have different spike length, the spike length, I think, is gonna affect the uptaken. So this is what we are studying now. So I don't have yet the results, but we are comparing the nanoparticles with the shorter spike with the one with the longer spike. So I can give you these results if you are interested. So drop me an email in like a few months, like it will be in January or February. And then the other question was why star shape cross better than spherical one? So it depends on the geometry that they have, that I have explained before. And what are the possible long-term adverse effects of nanoparticle as will be delivered to the brain? Regarding the silica, so the silica is going to degrade, and we don't have like, we are not concerning the long side effect about these type of nanoparticles. But then we need to think about the cerium oxide, because it's not gonna degrade in the brain. So we are also studying this part. And as I said before, we are monitoring the production of cytokine in the different type of cells. So we have started to monitor the pro-inflammatory and anti-inflammatory cytokine in PBMC cells, and we can see that some cytokine are like activated, but it depends on the concentration of the nanoparticle. So I just want to underline how the sites and the concentration of the nanoparticles affect the toxicity. I hope that I replied to all the questions.
Any other questions?
There are a few questions in the Q&A function. We've got one from Elias, and they're asking, is there any contradiction for nanoparticle treatment?
Any contraindication? Oh, we could think about this. Hang on, let me think. The answer is I don't know. So this is using different drugs, right? And do you see adverse effects? That we haven't studied this, to be honest. So I haven't seen any literature on this. I'm sorry, but I don't know. Have you read anything on it, Ali?
No, I mean, the contradiction can be related to the long-term toxicity of the nanoparticles, which it depends on the concentration. And also, I mean, with the nanoparticles, you can target the particles to the organ that you want to treat. So for this reason, you can like reduce the toxicity of these particles in another part of the body, in other organs. So this is the advantage that we have with the nanoparticles.
Do you expect in vivo CNTs to lose some efficacy in vivo formed by formation of coronas? Yeah, it's a good question. So Ali is, I don't know with the CNTs, Ali is working on this at the moment. You are putting the silica particles into the blood serum, right? Yeah, to test this.
I'm working. So now we are injecting these particles in the mice, And after like a perfusion, so we are collecting the blood and the organs. So we are going to harvest the organ as well, and we will do the quantification of the particles in the blood, and we will see also the formation of protein coronas, which is going to affect the activity of the particles. So we need to consider the formation of the protein coronas on the nanoparticles.
That's great. Thank you so much to both of you. If there's no more questions, then I just want to say a big thank you from IMSE to both of you for such an informative presentation, and also getting through so many of the questions from the audience. It's been really interesting. Thank you so much. So a recording of this webinar will be put on the IMSE YouTube channel very soon. And the next webinar in this series will be on the 18th of January at 4 p.m. And it's entitled Antibody Discovery versus Neurodegeneration. And it's presented by Dr. Francisco April. So once again, thank you so much to everyone who attended, and also to Dr. Pina and Professor Porter for giving such a fantastic presentation. Thank you very much.
Thank you so
Thank
much.
you.
Bye.
Bye.